So today, we're going to talk about Chapter 7, the MHC complex, which is, real classic immunology. And that's great. Okay, so as I said, today we're going to start Chapter 7, talking about some classic immunology with the MHC complex. And let me return to first, I want to note the Chapter 7 exclusions, which are listed here. I'm going to share my screen. So I'm making you read a lot this chapter, in that there's a because there's so many things that I didn't want to exclude, but it'll be worthwhile. So we will learn a lot. So starting out in the MHC complex, we touched on this already. The background for the name is that it's been known for a long time that skin could be transplanted between related mice, but not between unrelated, thus histocompatibility. Now, evolution wouldn't seem to select for this trait to stop mice skin being transplanted from one mouse to another. So, question arose: what is the real role of the MHC? And I should say, clever geneticists were able to find the set of set of genes that were responsible for this trait. And they call that the major histocompatibility complex. Ultimately, in modern times, we found that MHC molecules can present, are the molecules that present antigen to T cells. So on the surface, they're on the surface of cells, and if this is an antigen, my hands are up holding the antigen out for T cells, saying, look at me, and thereby asking the T cell to determine whether that cell is okay or needs to be attacked. Okay, so that's the function of the MHC complex. You recall that unlike B cells, T cells only recognize fragments of antigen associated with or presented by the MHC. And these are fragments that are derived from whole proteins broken up into peptides. So they've been broken down within the cell and then transported to the surface with an MHC. And the book cites work in the past from Benacerraf, which we won't talk about, and Wiley, and then also an experiment, a classic experiment by Zinkernagel and Doherty, which we will talk about. Okay, so class 1 and class 2, we see here in this slide, they're both on the surface. They both present antigen to T cells. In fact, they look very similar despite the fact that class 1 is made up of one chain, two chains, one much bigger than the other, and class 2, two chains that are approximately equal size. Class 3, we will kind of gloss over, are another set of genes in that locus, and they're very different. So class 1, 45 kilodalton glycoprotein. The alpha 1 and alpha 2 domains are most exterior in the chain, and they account for peptide binding. The small protein associated, it's called beta 2 microglobulin, and it's non-polymorphic. We'll see soon that the other MHC chains are highly polymorphic, among the most polymorphic, not the most polymorphic in the genome. So alpha 1 and alpha 2 domains form a floor with 8 anti-parallel strands, back and forth, flanked by rims of alpha helices. And, well, switch to it now. I'm going to return to this slide. So many of you have heard biology teachers ad nauseum talk about structure and function, right? Maybe too much now. And maybe me. But this is one of the best examples of structure and function where people saw a structure and said, aha, okay, this is what we like, you know? Not where you have to be convinced of something. So when the first crystal structure of first it was a class 1 with peptide, was done by Pam Bjorkman and Don Wiley in, like, I don't know, 1986 or something like that. It was a big thing that people noticed. I had some difficulty with pulling up the structure program that I use here, but I've saved a few of these still pictures from the molecular graphics. And on this slide that we see, you see on the left is the MHC molecule. The T cell receptor 2 chains is on the right. Ignore that for the moment, but focus on this. And here we see the peptide on end here, cradled in the MHC molecule. Okay. So I think this is one of the best examples of structure and function, and it really jumps out at you. So the T cell looking down, or I guess it's looking left at it, sees its ligand as both the MHC and the peptide. Okay, so that's kind of cool. I put the link in on the home page, and it showed the whole figure, which I guess is okay. Here's another drawing here. Here's another showing the side chains, and of course there's not these big gaps here, but you see again the peptide is in yellow, making intimate contact with the MHC molecule, and because this also is later they were able to get structures of TCR MHC peptide, and we see them all making close contact, as you might express, for a good interaction. Okay. So I hope you share my enthusiasm of that. So this is whatever, you know, ribbon and crayon structure, but this is really cool to see that, especially the end-on view like this. I don't know if you guys are aware of this or if Jackie presented this to you in biochemistry. I've used the program CN3D, letter C, letter N, number 3, letter D, for a long time, and I know Jackie is familiar with one of the chemistry-based programs, which I didn't know was so good for proteins, and I think she's probably right that it's easier to use, but I at one point achieved a certain amount of skill working with this one, and I fooled with it a bit. But I think it's really cool in some cases to look at these structures, and you can do a lot of things. Like you can, in this case, each chain is a different color, and you can do something like ask, where is residue 309? You know, highlight that, and you can relate position in the linear order of peptides with a position in the three-dimensional structure. But anyway, so this is cool. Structure is kind of a cool thing. And so this shows you well how the peptide is cradled in the groove. And of course, you saw in the other picture how close together things are. So it's not surprising to learn that there's contacts made between peptides and the MHC, whether that be class 1 or class 2. So in the original crystallography, they saw peptides in the groove, and turn to slides here. And it turns out that when they were able by biochemical techniques to look at peptides in the groove and found a lot of self-peptides, which was kind of a surprise at the time. Class 1 and 2 are part of the immunoglobulin superfamily. Remember we said a lot of them, these are. The alpha-3 domain interacts with CD8 on T cells. There's a mention in the book of, I think this edition has it as well, a cell line called Dowdy that doesn't make DAUDI, that doesn't make beta-2 microglobulin. In those cells, class 1 does not come to the surface. That's a general rule in immunology. And as soon as I make a statement like that, there'll probably be examples that aren't true. But in many of these complexes, like the T cell receptor with all the CD3 chains, or in this case, class 1 with beta-2 microglobulin, if they're not both there, they don't come to the surface. That's a good general rule. So let's see. Class 2, I'm going to move down to the next slide. Here's class 1 again, some of the things that I talked about. The cleft forms an 8 to 10 amino acid, binds an 8 to 10 amino acid peptide fragment from an antigen. Oh, I didn't get a chance to say one of my other favorite things to present. The structure you saw before has been likened to a hot dog in a bun. So I love that because you'll remember that. It's goofy and you'll remember that. So hot dog, bun. Beta sheet on the floor, 2 alpha helices on the walls. Associates with beta-2 microglobulin. The interaction with between the beta-2 microglobulin and MHC class 1 is non-covalent. Okay, class 2, also of course a member of the immunoglobulin superfamily. And it's interesting, we see here that the 2 molecules, 1 and 2, are almost superimposable, which is kind of remarkable because there isn't that much sequence homology. And in fact, one complex has 2 similar sized chains and the other has one much bigger than the other, but yet the 2 structures, the 2 complexes are almost superimposable. So again, a heterodimer 8 strands of a beta sheet, 2 helices. Again, the class 2 peptides are larger, so the joke is that the hot dog sticks out the end of the bun in class 2 and not really in class 1. There's some variability, 13 to 18 in general. And I'm going to skip through some of these questions shown here. And this slide makes it clear about the superposition of the MHC class 1 and class 2. And we see in B the class 2 with a larger peptide here, where the ends are more closed around class 1. So variation MHC molecules, another thing where you've all heard about worrying about transplants and people being able to accept transplants and meeting close relatives. So there is great variation, polymorphism in MHC, more so than any other genes. Many allelic variants in humans. Also, there is more than one class 1 gene, more than one class 2 genes. So that makes sense. I've always had a little struggle with this, that it makes sense from a population perspective that there is great variation protecting us against a wide variety of pathogens. On the other hand, selection operates at the level of the individual, so I've had a little trouble rationalizing that in my mind, but anyway, there is great variation. And there are people who are extreme nerds on this, and they know that, say, of a certain ethnicity, they know that these alleles are common and there's this percentage and that kind of thing. There's people who really know that, and that's really practical too. Yeah?
When you're talking about the polymorphism, like I understand there's variability, but like how is it variable? Is it variable in size or like the rate of presentation?
What they bind for one.
Yeah.
Yeah. Well, that's the next topic. Yeah,
Okay.
we're getting right there. Yeah. So, how does this limited group of molecules present a vast, diverse array of possible peptide fragments? Well, a given molecule can present numerous peptides. This is sometimes referred to as promiscuity. So there's a lot of humor in some of these things. And some peptides can bind to several different MHC molecules. When I worked on this kind of stuff, we worked with a peptide that would bind multiple MHC class 2 molecules, and that was part of the reasoning that you could substitute and that you could mix and match different ones. One common feature of binding is that peptides are always in pretty much an extended conformation. Maybe a little bit of a kink, but an extended conformation. So the K subscript D for these peptides, they vary from about 10 to the minus 6 to 10 to the minus 10. So most can be bound in an in vitro situation. So where do the class 1 peptides, the peptides bound to class 1, come from? So usually these are endogenous proteins degraded in the cytosol. And by endogenous, I mean self proteins that are degraded. There's continuous degradation, recycling of proteins using a proteasome. Probably touched on that in cell biology maybe, or one of the classes. And also when I say endogenous, that includes viral proteins when the viral infection with, coming from within the cell, or even a bacterium that has been able to infect and get inside the cell. So we say endogenous as opposed to being taken up from outside. Okay? So they interact with class 1 in ER. So the book says something like there's 100,000 copies of class 1, so many different peptides can be presented, but it's different for different MHC alleles. So here we see, in some of the cases, there's a slight bulge in the peptides, but it's class 1. But typically, in the extended conformation, for MHC class 1, usually 8 to 10 amino acids long. Anchor residues, anchor peptides in the groove, and typically they have consensus sequences that work, not usually just one amino acid, but it might be the case that, say, a big hydrophobic amino acid is absolutely required at a particular position.
Is there a mistake in the textbook? Because I don't think hydrophobic residues will form hydrogen bonds. Hydrophobic residues should form
Yeah.
van der Waals forces.
You don't think they mentioned hydrophobic residues?
They did,
Oh.
but they wrote here hydrogen bonds, which I
They don't hydrogen bonds? Well, they don't typically not, although often there's hydrogen bonds between the outward facing amino acids and the T cell receptor. So, and I guess in some cases, there are hydrogen bonds. I think not all the interactions between MHC and peptide are hydrophobic. I think there are some hydrogen bonds as well. But I think the anchor residues especially tend to be hydrophobic.
Yeah, hydrophobic then it's not hydrogen.
That's right. So here's an example of, it's interesting, they say, they don't cite it, they can cite real data here. And some anchor residues here for this nonomer, and we see here in position 9, hydrophobics, LL, leucine, isoleucine, and leucine, and consensus at position 2 with all glycine, and position 3 with all proline. Okay, whereas a different, in their example, which I wish they'd say what it was, here we see with a different MHC, this variant, tyrosine Y at position 2, and position 9, isoleucine, valine, or isoleucine. Okay, so these ones sit in hydrophobic pockets in the class 1. So what this allows people to do is make some predictions about, for a given protein, you can predict what might be correct epitopes, what might be epitopes that will bind a given MHC. So I think it's in italics in the book, it says the set of class 1 alleles inherited by each individual will determine which set of peptide fragments from a larger protein will be presented. So in a given protein, it's not the case that every possible 8-mer or 9-mer would be presented at all, or presented by a given MHC molecule. Okay. And the book cites data from one study with finding 2,000 peptides from 2 MHC class 1 molecules. It's estimated that each complex is present 100 to 4,000 times, and in some studies they suggested that even one can trigger a T cell. So, variation in the genes and proteins of the MHC molecules in determining what peptides can be presented. Peptides presented by class 2 proteins. These are usually derived from exogenous, meaning extracellular processed antigens, so debris from the media, something from outside, perhaps phagocytosed. So the groove is open at both ends, allowing longer peptides, relatively constant elevation. I think the interaction is governed by more amino acids typically in class 2 than in class 1. So one can predict binding, as I said, by knowledge of anchor residues. In class 1, the pattern is anchors at either end and a bulge in the middle. Of course, amino acids whose side chains are pointing upward, away from the presenting cell, are directed towards the T cell and towards TCR, and they allow for interaction with the TCR. MHC class 2 presents peptides to CD4 cells. Again, there are consensus sequences, but the influence of anchors in MHC class 2 is not as strong. So MHC molecules must be able to present a wide variety of epitopes, and they do this by promiscuity, as I said. A different tact than the antigen receptor genes, which have all this variability by different rearrangement. So I was thinking at this point of doing a little experiment and scanning a protein for a possible epitope. So I'm going to take as a model protein chicken ovalbumin. This has been used a lot in immunology, and I'm especially familiar with it because the guy that was kind of the major immunologist at the UW is the one that really developed this system. And this is chicken ovalbumin as an antigen. And you might say, why chicken ovalbumin? Well, I think there was an intention to avoid viral proteins because, well, different mice or people might or might not have been exposed to a different virus, but probably no mouse or no person that might be studied would have been injected with chicken ovalbumin. Okay, so that's why they did this. Oh, and also they can make a lot of it. So let's look at here's a particular MHC molecule. Talk about this in a minute. And it has a motif that recognizes so this is a motif that is common for this MHC, hydrophobics, with a 0 here, X0, XX0. So thought I would turn to this. So on the home page, there's a link, it says Unipro to ovalbumin or something like that. So I encourage you to look that up and scroll down and you will see a protein sequence. So this is the protein sequence for chicken ovalbumin. So I encourage you to spend a couple minutes, I don't know how adept you are, just to look for a situation where you see hydrophobic, space, hydrophobic, space, space, hydrophobic. This may not be your favorite thing to do. Yes, you already found it?
No, no, that's not it. Is this something you're gonna want us to be able to like do? Like on a quiz or test?
Well, I don't think it's unreasonable to ask that, but I probably wouldn't just be. But, I mean, I guess that'd be really testing if you know what a hydrophobic amino acid is. Big hydrophobic too, I should say, for these guys. Do you want to know? Are you confident enough about those? Yes.
103 maybe?
103. Okay, let's look at that. So starting at 103, so that would be A blank R. Is that what you're thinking?
Yeah.
So R is hydrophilic, so that's not ideal there. There might be other, I mean, there's one that's widely known, and there might be others that are sounding a good match. But mostly I just want you to get the idea what people do, not what the answer might be. So you can predict possible. You got it, Stacy? No?
Maybe. How about 19?
19?
Yeah.
Okay, so L.
L K V H H A. Ah no.
Okay, V. H A N. So I'm forgetting the actual, so N is not hydrophobic, although it's big. So, well, I'm saying I know one that isn't widely used that does work, and it's not that I can say that other things don't work at all. Anyway, you get the idea. Do you want another minute to look at it? Yes? No? I'll give you another minute. Get the idea what people would do. At one point, people would also synthetically make peptides spanning a small protein and then see which of them would work for a given MHC.
Can you give us a 50 base pair range? It's kind of long, yeah.
Sorry, where?
Can you give us a 50 base pair range?
Oh, okay. Yeah. How about looking between 250 and 280?
Okay, we can do that.
You can kind of make a word out of it. Well, I mostly just wanted to give you the idea of what you would look for, given a certain motif. Anybody close? So it's right here, Sinfecal, SIINFEKL. So that's a widely used system by Mike Bevan's lab, SIINFEKL, presented by K of B. Okay. So just give you an idea what people can do in fact do. So that in fact is an immunodominant peptide, is dominant in the mouse immune response. Okay, Synvecle. And again, I think everyone, immunologists, is fairly familiar with it, but especially people here in Seattle, because Mike Bevan was like the major guy at the, I think he's retired now at the U, but that was a system that he works in. So kind of like everybody has some familiarity with the lac-operon for gene expression. This is kind of like the lac-operon for people who do class 1 work in the mouse. Okay. So this system allows for a large number of epitopes to be presented. Again, remembering that a given MHC will present a large number of peptides. And originally, work with MHC was done with transplantation. It's found that the MHC locus encodes 3 major classes. The MHC locus is on chromosome 6 in humans, on chromosome 17 in mice. It's called HLA in humans, called H2 in mice. MHC is a word that can be used for all, a label that can be used in all species. Okay. 3 sets of classical MHC genes. There's a comparison of peptide binding by class 1 and class 2. Closed versus open, size, amino acids, anchor residues versus one called conservation. So note those differences. Okay. So here, next slide shows organization and inheritance of the MHC genes. And we see class 2 together, class 1 here. Class 3 are proteins that are related to the immune system, but they don't have the great diversity, and I won't discuss them anymore as far as their genetics. So I had joked when I used to do this kind of stuff, or when I was first learning it with my boss, and I think I said something like, okay, so they have an MHC nomenclature meeting every few years just to confuse people when everyone starts to catch up with knowing everything, and then they try to confuse people again because there's really a lot of lore and a lot of difficulty with it, in my mind. Okay. We see here in the mouse 3 class 1 genes, G, L, and K. The last study we did was in the K of B, the B haplotype. In the human complex, the class 1 genes are A, B, and C. And the class 2 genes are given these names, DP, DQ, and DR. Yes.
In the human HLA complex in one, what is the difference between HLA, B, C, and A?
Why do they divide class 1 genes into 3 groups? Is that what you're saying?
Yeah. What's the?
I think the different locations and also different different alleles of A are closer to each other than a B.
Okay.
Okay. So it's also so-called non-classical class 1 and class 2. So this has been a big area of research when people first began to have ability to look at genomic, ask questions at the level of genomics. skip through this. So simplified map of mouse. So you're saying I needed a simplified map, right? You're saying none of this is simple. So we see this is a large area of the genome, 1500 kb in the mouse, again on chromosome 17. And we see some of these genes here, IA and IE in the mouse for class 2. Again, in the red, the class 1. Human HLA, class 1 in the red again, and the light blue here in the class 2. Okay. So class 2 is IA and IE in the mouse, and DP and DQ and DR in humans. There also exists non-classical class 1. Some play a role in self, non-self recognition, for instance, HLA-G. Sometimes there's more than one gene. There's more than one DR locus, and different haplotypes have different loci. Also, this allows for different alpha-beta combinations. So diversity, 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 right? In class 2, both genes are polymorphic, whereas in class 1, beta-2 microglobulin is non-polymorphic. In class 3, there's TNF alpha and beta, some complement genes, and lymphotoxin. There's also genes called DM and DO, which play a role in class 2 in antigen processing in class 2. Okay, I touched on this yesterday, that allelic forms of MHC genes are inherited in linked groups called haplotypes. So if on one chromosome you have this allele and further downstream you have this allele and this allele, those tend to be inherited together. There is very little recombination within MHC. And I didn't check this in the book for this current edition, but it seemed that they really were reluctant to make a strong statement here, and to the point of almost being wrong, that of course at a given locus, in a given meiosis, there's going to be not much crossing over, but with time, there's going to be randomization, right, of with many generations between different loci, right? Does that make sense? But there is a much lower depressed level of recombination in the MHC locus than would be expected. So that means these tend to be inherited together over long periods of time. Okay. So that's been sort of useful in our knowledge sometimes. If you know sometimes one allele at human DR, then you know the allele likely at DQ, but it's also made things somewhat different at times. Okay, so there's great polymorphism at the locus. Genes are inherited as a group. There's very rare recombination. This reflects a genetic phenomenon called linkage disequilibrium. Have you heard that term? Okay, good. I'll write that down. Linkage disequilibrium. And it's a operational you would expect the frequency of two alleles being together to be dependent on the frequency of each one individually and being multiplying them. But if two things tend to be together, they're said to be in disequilibrium. This might happen because of a disease gene that's evolved relatively recently, and that in humans that relatively recently could be several hundred years ago. And then it's going to tend to be inherited with the markers that were near it when it first arose, okay? Because it takes a while for randomization, right? But this phenomenon of linkage disequilibrium exists in the MHC locus for long periods of time because recombination is rare. Okay, so that's a bit of a genetic term here. So these link groups are called haplotypes. Again, sometimes you hear haplotypes referred to when following a disease gene, and which a de novo disease gene, which is on with a certain set of others, and then it's going to stay linked to that for a while, because it takes a while for there to be equilibrium. And that's how you see haplotype used sometimes, but it's used here as well. So each individual inherits one haplotype from each parent. The inbreeding of mice, as I talked about Monday, is a way to control all this variability. So we talked about how this variability is great for the species as a whole, but sometimes we want to have a handle on what we're doing. It's good to have these inbred mice. And speaking of mice, so syngeneic mice are identical at all loci, and congenic mice, which people make use of, are genetically identical everywhere but a particular place that you're studying. So in this inheritance, offspring get one haplotype from each parent. So because humans are outbred, sounds like a weird thing to say, we're outbred. So people are almost always heterozygous at each locus. Mice have been inbred, so they're homozygous. Okay, well here are some haplotypes, and haplotypes are given as letters that are presented as superscripts. For instance, a few minutes ago, we talked about epitopes for K of B. That's how you say it. The K locus, B haplotype. Okay. So here are some different alleles. D here for the whole, or different loci. D here for the whole class 2, and for class 1, K and D, and then A and E for class 2. And sometimes for the mice, people have intentionally tried to create strains that were mixed to localize an observation, the genetic basis for an observation. But most of these classic strains are, of course, have one haplotype. So some of the well-known ones, yeah.
Oh, I didn't mean to interrupt.
That's right.
So, like, I work with a strain of mice that's black 6, so that's C57BL.
Yeah.
And we've bred in HLA-A2 to that. Would it then replace one of these, or would they somehow engineer it to
unless
be
they've
an
done
addition?
some effort to do
Okay.
that. Yeah. Well, sorry, did they introduce like by a transgene or by conventional breeding?
I think it's transgene.
Okay, it's by a transgene and it wouldn't replace it.
Yeah.
Okay, so some of the well-known strains that you all know and love or have worked with, the B6 mice have the B haplotype. And the BALB-C mice have the D haplotype. So some of those are shown there, and there are various recombinants as shown here on the slide, B10 .A2R. So there's a lot of nomenclature. There are people who are really into this and know everything about this. So I'm not one of them. And of course, different strains, mouse strains, as we saw Monday, they don't differ only in this. Some of them are more prone to certain kinds of tumors. Some of them are more prone to certain kinds of autoimmune disease. And that reflects that's why people use different ones of them. Also, in some cases, they then have to do crosses. They make an observation in a certain strain, and they want to see if they want to tease out differences there. They might want to put that phenotype with a different MHC, for instance.
Can
Yes.
you please explain what is the K superscript B?
So it would be K of B. That would be, yes, if you were when I was talking about. So like that.
Yeah, what is it?
What does it mean? Okay, that's the locus K and the haplotype B.
That means that locus K is the.
Yes, that's the B. And just, I think it's hard to just throw out information, but it's good to memorize some specific things. So the O'Vallivan system, that's a K of B restricted epitope. Synfecal. See, it helps that if it's something you can try to pretend is a word. So synfecal, easy to, rolls off the tongue, right? Okay. So this is a cool slide. So these genes are codominantly expressed. It's important to note this because it's different than the antigen receptor genes, which exhibit allelic exclusion, where once one has successfully rearranged, then the other one, the other allele, does not rearrange. So it's important to note these are codominantly expressed. So here we see in this figure, from the maternal MHC, 2 forms of K, 2 forms of D, 2 forms of L. I'm sorry, from the maternal is 1 form, but 1 from the paternal. And similarly, you see these different forms in class 2. And note that because class 2 is polymorphic in both alpha and beta, you can theoretically have even more diversity. So what do we like in immunology? Great diversity, right? So it says both parents' genes are expressed in offspring, best chance to have some capability of presenting all possible antigenic peptides, but it makes transplantation more difficult. And some of this stuff was worked out in inbred mouse studies. So there's great diversity at both the individual level and the species level. Alleles differ among individuals. So flexibility for unexpected changes now in the future. So here's a discussion of HLA haplotypes here. And so this says a typical human family. Here with 1 parent being A/B, 1 being C/D, and having all these possibilities. And here they do show a recombinant here, even though that's going to be rare. And so I have my notes here. Not only are the genes expressed codominantly, but functionally they're codominant. Okay, let's look at some real experiments here. And we're working with our mice here, our red and yellow mice. They're actually not really red and yellow, but in here we have mouse strains, a B MHC strain and a K MHC strain, and so the progeny will all be B/K, written like this. Now the question here is, how about skin transplantation, that favorite, that favorite crude tool of the immunologist? So if the B parent, looking here on the right upper, if the B parent is a donor to the skin to another BB, then it's going to be accepted, but to a K, it's not going to be accepted. On the other hand, progeny B of K can accept that B graft, because B is recognized as self by that offspring. Similarly, the offspring, the BK offspring, can accept the graft from the K parent, but the K parent cannot donate the graft to another BB. So, yeah.
So it functions the same way as blood groups?
As what?
As blood groups? Like type A, type B blood. Does this
Oh,
function similarly?
I guess so in this respect, yeah. Okay, so the BK progeny cannot donate to either parent, but can to another BK. Okay, so a little bit about inheritance of the haplotypes. Okay, so that's kind of cool. So progeny are histocompatible with both parents, but neither parent can receive graft from the progeny. So it's rare for two unrelated individuals to have the same HLA, hence the difficulty in finding donors. Here's some numbers about diversity in the human population. And so here we have these different loci, A, B, and C being most important, number of different proteins known. And we see DP, DQ, and DR, each with alpha and beta. So again, great diversity here. You see region is blank. What is that? Polymorphic? I'm not sure. Okay, next. So variability within the MHC molecules, many different alleles. You might ask, where are these differences? And these differences are at the business end, where you'd like them to be, where they make a difference. So we see here, this shows the sheet as a floor and the sides helices, and we see that in the red parts are places that are highly polymorphic. So they're polymorphic and that has a functional difference, different peptides, and they're able to bind. So, great diversity at both the individual and species levels, multiple alleles at many loci, and then each allele can bind many peptides. And you note the possibility of trans alpha beta pairs in class 2. So great diversity in HLA, and many alleles may not yet be known. Sometimes people, there's a whole thing about finding isolated ethnic groups that might have yet new alleles not known, and people have tried to do anthropology looking at allele frequencies, say trying to look at the settling of the Americas by allele frequency. So that's a whole area in itself. MHC polymorphism improves resistance to disease. Of course, different abilities to bind different peptides. Focused in the antigen binding groove. Okay. So recognize that these differences are functional. Also, an interesting sidelight that there's a box in the book about class 2 differences in autoimmune disease. So, you're familiar with many autoimmune diseases: type 1 diabetes, rheumatoid arthritis, multiple sclerosis, many others. What's the gluten sensitivity? I'm blanking on it.
Celiac disease.
Celiac, yeah, okay. Number of others. For these and many others, there's an association between susceptibility and alleles in class 2. It's a little bit difficult for people to understand this, and it's because when you think about a normal gene, if you think about sickle cell disease or cystic fibrosis, we think about a particular allele, and if you have it or if you have 2 copies of it, you will have the disease. In this case, it affects susceptibility. So just means if you have a copy of the allele, your susceptibility is significantly different. But it doesn't mean you always will get it. And partly this is because there are genetic factors and environmental factors for all these autoimmune diseases. And of course, this is being teased out. And in some cases, there are genetic factors that are beyond the HLA locus. But this has been a big source of interest. And different loci are important with different of these diseases. People try to figure out what's responsible here, and it's just without much success, really. So, so various class 1 and class 2 alleles are associated with an increase or a decreased risk of autoimmune disease. So genes and environment are important. There is the idea of relative risk. I don't know if you know that, relative to the general population. So you get numbers for something like IDDM or RA. One DQ allele gives a 5.6-fold relative risk for diabetes. Another allele is protective. We mentioned earlier that these genes, alleles, tend to be inherited together. So therefore, especially in early studies, it was hard to determine which locus was actually responsible, since this locus would always be, the allele of this locus would always be present with a certain allele at this locus. Okay. So for something like IDDM, the haplotype, so early on, before we could sequence whatever genes we wanted, the MHC regions were defined by antibodies and given DR numbers, so like DR3 or DR4 for haplotypes. So those were antibody-based designations. And really, people didn't know if the genetic susceptibility was due to DQ or DP or DR.
Oh, the DR doesn't refer to DR.
DR what?
The DR number doesn't refer to the locus DR.
Yeah, the DR was used, the numbers were DR using antibodies, and DR is a gene now, and the DR gene products are expressed, I think, like tenfold more than DP or DQ, so it's most likely most important. But, just a little anecdote, so the institute where I worked in downtown Seattle for many years, the guy who was head of it, his claim to fame was that he had worked with the few recombinants found between DQ and DR, and he had found that the greatest contribution towards genetic susceptibility was at the DQ locus for a certain haplotype. So that's the kind of thing people do. Okay. All right. I'm going to keep going for a bit. So, so originally people just were able to associate haplotypes with susceptibility or protection. But then they began to be able to determine more what actual gene. So the attractive possibility that susceptibility alleles might be the only ones that would bind a particular epitope or that don't bind has not proven as fruitful as one might think. You know, it's not the clear, obvious experiments have been done. So it's still not really clear what's going on. Okay, any questions about that? So it's a good area, human differences and susceptibility to autoimmune disease. I think, you always run into trouble when you say always the case, but I think it's almost like a proof that something's an autoimmune disease if there's an association with class 2. A long time ago, I heard a talk where someone said that for these class 2 autoimmune diseases, that women are more susceptible than men for all of them. Although in some cases it's really close to 50-50, and in some cases like lupus, it's really strong bias. And at one point there aren't as many associations with class 1, but one of them is ankylosing spondylitis for class 1, and that's more common among males. I have no idea what that means, and I don't know if anyone knows. But that's lore. There's a lot of lore about this stuff. Okay? Any questions about this stuff, about susceptibility? Okay, so class 1 proteins, what do? They present peptide, but they present one thing that was a surprise, and this was discovered, people at some point, maybe early 90s, found biochemical techniques to strip off the membrane peptides and proteins that weren't tightly associated, then specifically strip off those that were associated with a particular MHC. And when they found a bunch of peptides, they could do this big time sequencing with mass spec and sequencing, and they could look at peptides bound by a particular MHC. And to their surprise, there were a lot of self proteins. So this provides a way of checking that cells are self and are generally healthy, but also this display, yeah.
Sorry. When you say self proteins, do you mean proteins from within the cell?
Yes.
Okay.
Yeah, and I guess that is, and we really do mean encoded by the self genome in this case. So, but also these peptides can be different, and it could be used to show infection from a virus. Okay. And so therefore, CD8 T cells are alerted, and these cells can be targeted. We'll talk about this later, but the absence of class 1 can actually alert natural killer cells that look for the absence of class 1. MHC class 2 proteins, as we've learned, are on a subset of cells. They mostly present extracellular antigenic peptides. The expression is more restricted to so-called professional antigen-presenting cells. So This helps direct responses towards these other threats and activates helper T cells. So the book suggests that fewer cells need to express class 2, thus only professional APCs do that. So together, these guys work together, class 1 presenting intracellular, whether that be from viruses or bacteria that have been taken up, and class 2 extracellular. Sometimes there is a phenomenon called cross-presentation where class 1 presents extracellular. So, yeah.
So, just to clarify, so this one, there's like a smaller quantity of class 2 proteins? Because when we were looking at the list of haplotypes,
Yeah.
there were a lot more haplotypes for class 2.
More different alleles?
Yeah,
I think that's probably true.
okay. So there's fewer of them, but greater diversity?
Oh, you mean the total level of protein on the surface?
Yeah.
I kind of think there's more class 1 than class 2, but I
Okay.
wouldn't swear to it. Okay, so MHC molecules are important for development, tolerance, and development of mature T cells, and they present self-peptides as well as foreign. And the presentation of the same peptide can have different effects depending on when and where presented. For instance, during development of that T cell in the thymus or later in the periphery. So we see class 1 on all nucleated cells, class 2 on only antigen-presenting cells, such as macrophages, B cells, and dendritic cells. Although they're expressed, class 1 is expressed everywhere, it's not the same amount each place, and in the text says an estimation of 0.5 million MHC per lymphocyte, 1% of membrane proteins, but notably fewer in the liver, for instance. Maybe the reason for relative success of liver transplants. I knew someone once who was involved with liver transplants and said, although they try to do these MHC matching, sometimes it almost seems like it's not that worthwhile. And some neurons in some stages of sperm cells seem to lack MHC class 1. So many different alleles, many different self-peptides on class 1 of a given cell. If a cell is infected, then the pathogens epitopes, some of them will be presented. Different individuals with different MHC will present different peptides. Note the importance of constitutive expression of class 1 in that NK cells can kill cells lacking class 1. And note, of course, the continuing war between pathogens and the immune system, that it's not surprising that some viruses have discovered ways to downregulate class 1, sometimes by making a protein that binds to class 1 so it doesn't come to the surface. So our immune system has evolved a way to recognize cells that don't have class 1, and natural killer cells can kill those. Class 2 expression is restricted to antigen-presenting cells. These professional antigen-presenting cells have the ability to deliver signals through co-stimulatory ligands. Some of these do this are dendritic cells, B cells, and macrophages. Dendritic cells are constitutive for high-level class 2 and co-stimulatory molecules that makes them really good at starting immune responses. Macrophages need to be activated before they make high levels of class 2 or co-stimulatory molecules. B cells constitutively make low levels of class 2, but not co-stimulatory molecules, but they have a special role with their cognate antigen, the antigen that their immunoglobulins recognize that they can then take up. MHC expression can change with changing conditions. So some transcription factors, C1TA and C2TA for C1TA for class 1 and C2TA and RFX for class 2. Those people who have genetic deficiencies of RFX, a transcription factor, or C2TA, transcription factor, have something called Bayer lymphocyte syndrome, which is a clinical problem, but it also provides cells that are deficient in MHC for people to add back MHC in transfection experiments. Viruses sometimes shut down class 1 expression. Cytokines can upregulate MHC expression, especially interferon gamma, interferon alpha, sorry, and TNF alpha. And I mentioned earlier that some viruses interfere with class 1. Cytomegalovirus binds beta-2M, so class 1 can get to the surface. Adenovirus blocks proteins that transport peptides. And it, in your book, it's noted that everyone's favorite model organism, the Tasmanian devil, is prone to a cancer that decreases class 1 expression. Class 1 expression is increased by interferons and TNFs from innate immune cells like phagocytic cells. So interferon gamma upregulates C2TA and then therefore class 2. Corticosteroids reduce class 2 expression and depress immunity. So class 2 genes were originally isolated as, or defined as IR genes or immune response genes because in some model systems they control the immune response. So what might be the reason? You could logically think either the binding of a particular peptide or maybe holes in the TCR repertoire. Which are these? And there's some evidence for each. So MHC and immune responsiveness, and these two ideas, determinant selection, ability to bind particularly processed antigenic peptides, or holes in the repertoire. Maybe those T cells that detect certain foreign antigens will be eliminated during thymic development. And both seem to be partially correct. When you think about, in the real world, response to a given pathogen, though, remember, there are many proteins on the surface of, say, a bacterium. And there are also, typically, even in one protein, there are many possible epitopes. And so while different MHCs may present different epitopes, the idea of the completely, this protein can't be seen or this pathogen can't be seen at all doesn't really happen very often in real situations. Okay, so heading in the book is seminal studies demonstrate that T cells recognize peptide presented in the context of self-MHC alleles. And so people realized in the 1970s that T cells only recognize antigen in the context of MHC. Originally people knew that both were important, but we saw the figure of the peptide being presented by the MHC, but before that it had been conceivable that the MHC and the peptide were different as far as how they were presented to the T cell. So also the MHC of the T cell and the APC must match. So of course this occurs normally, naturally in the unmanipulated host. So we have this dual specificity of the T cell. In 1973, it was found that antigen presentation by a macrophage caused proliferation of T helper cells. That's how they initially, T killer cells kill, T helper cells proliferate when given antigen. Found that macrophages could cause proliferation only if class II matched. So this led to a couple of seminal experiments here, and classic experiments, and I'm going to present them now and then let you go. But these are really important experiments. I know you wouldn't want to leave without hearing them. So the idea of MHC restriction, you may have heard that term. So restricted to recognizing peptides that are presented in the context of self-alleles. 2 experiments were critical in this understanding. And I'm surprised they don't even give each its own slide. I think that's horrendous. So you might remember, I think this will be the first experiment I present to you, or that maybe anyone will, using guinea pigs as a model system. So here we have guinea pigs as of 2 types. One strain is not infected with antigen, or given antigen. Then the other is. After 7 days, they harvest lymph node cells as a source of T cells for the antigen treated guinea pig. This is an old technique for a column that retains macrophages, but T cells will pass through. So we now have antigen prime T cells over here. On this left branch of the experiment, these cells are grown this exudate is grown in a plate, in a well. Macrophages are adherent, they'll stick. So now we will mix now at this point, these are given antigen. So now they're antigen pulsed macrophages and mixed in with T cells. And what is observed is that the T cells and the macrophages have to come from the same strain. We see here that we get a plus result only if the macrophages and the T cells are both from strain 2 or both from strain 13 or from the F1 of 2 by 13. But if they're mismatched, T cells from strain 2, macrophages from strain 13, or T cells from strain 13, macrophages from strain 2, there is not this proliferation, okay? Yes?
What is the reasoning for using guinea pigs as a model in this experiment?
Why is what? Sorry?
Why would you use guinea pigs for this
I
experiment?
don't know. Probably, historically, some people are good at doing what they do. So this goes back to, this is probably like mid-70s, I think. And maybe, certainly mouse work was more advanced than guinea pig work, then maybe it wasn't quite as easy now. Yeah, there's probably some technical thing, I don't know. Maybe these guys, this was Shivat, and maybe they worked in guinea pigs. So this phenomenon came to be known as MHC restriction. So the T cells have been educated, they say, or undergone positive selection on self-MHC. They won't respond to whatever peptides might be generated in a different strain. Okay. Now, let's look at the other experiment here. This is known as the Zinkernagel-Doherty experiment. Zinkernagel is one of those names you remember. So, in this case, they made use of an antigen is a virus called LCMV that's been used a lot experimentally. One advantage of using an antigen is that a virus as an antigen is that it can, depending on the virus, it can spread throughout the body and cause a huge response. Maybe too huge in some cases. Okay, so let's look at this situation here. So here we have the haplotype K mice are infected, and after a period of time, spleen cells are collected. We can expect that these will have T cells, including T cells that will kill target cells. So these T cells from the infected mouse were mixed with target cells that are the right haplotype, H2 of K, or H2 of K infected cells, or infected cells of a different haplotype. And the observation was that there was killing by the T cells only if the targets were both infected with LCMV, therefore having antigen, and also having the right haplotype. Okay? So the H2 of B infected cells, there was no lysis. This technique for killing, it's called a chromium release assay. Don't ask me how they came upon this, but apparently it's easy to osmotically shock cells to take up chromium-51 so that then if those cells are killed, it releases that radioactive chromium. Classic way to measure killing by killer T cells. Okay, so note here that the H2 of B infected cells are not killed. So we need both the presence of the antigen, the infection, and the right MHC. This is a classic experiment that demonstrated the restriction of T cells, restricted to the right MHC. Okay, so Zinkernagel and Doherty, Nobel Prize won for that MHC restriction. I wasn't as aware of the other experiment when I was not teaching, and maybe they just had bad PR or something. I'm not sure. Maybe there were some other elements of it, or maybe people were prejudiced against guinea pigs. I don't know. But especially the second experiment should know that, and the protocol and the results and what it shows. Okay, and we know now the explanation for that with the crystal structure that the T cell receptor recognizes the compound ligand of both peptide and MHC. Okay? Well, I hope it gave you enough today. Get your money's worth. So I'll finish with that. But that, especially at the end, is a classic experiment. So I shall stop at that point. Any questions about that last thing or anything about MHC? Okay, very good. Well, we will continue with an async lecture on Friday. I can't promise this much excitement though on Friday. This is really Zinkernagel and Doherty is. Okay, good.